Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. Introduce my name is Farah Salsabila Nuranti. I will explain about DNA. Let's go. What is DNA? Every living thing has billions of cells forming itself, starting from skin cells, blood cells, and brain cells. Although different in shape and size, each cell has a part of the nucleus. Called the nucleus, it is in the nucleus that DNA is located. DNA, or deoxyribonucleic acid, is a molecule that stores genetic instructions in every living thing. DNA molecules are molecules that are constantly composed of repeating similar groups called monomers. The monomers of DNA are called nucleic acid, which contains of a derived sugar group as the base group, a phosphate group at atom number 5, and base group at the first carbon atom. The nucleic acid of DNA are called deoxyribonucleic acid. DNA has a leader-like structure. Each rung stores genetic information in the form of 4 types of base molecules consisting of 2 groups, namely purine and pyrimidine, each of which consists of 2 types of molecules, namely A for adenine, T for thymine, C for cytosine, G for guanine. Each one purine molecule can pair to form hydrogen bonds with one pyrimidine molecule. Cytosine will pair with guanine with their hydrogen bonds, and thymine will pair with adenine with 2 hydrogen bonds. If there are 2 strands of DNA that are aligned or at the most complementary to each other, it will form a secondary structure in the form of a double helix. DNA replication is the process of DNA double that occurs in the nucleus and occurs in the synthetic phase. In the interphase in cell division, there are 3 models in DNA replication, namely conservative model, where the 2 parents DNA strands serve as templates. Semi-conservative models, where the 2 parents DNA strands separate and each makes a new standard as a complement. Conservative model, where several parts of the parent's DNA are scattered as a template, then each part makes a new DNA part as a complement. Enzyme used in DNA replication. Helication, breaking hydrogen bonds to open the DNA double helix chains into 2 single chains. RNA primase, forming primary RNA as the beginning of the formation of a new DNA chain. DNA polymerase, combination nucleotides into a long DNA polymer. DNA ligase, contains DNA fragments into a complete DNA standard. In theory, the DNA double helix thread is opened into two single strands by the enzyme helix. With the help of topoisomerase, which reduces the tension of the DNA strands. The single DNA strand is attached by single strand binding protein to prevent it from reforming the double helix premise. From RNA oligonucleotide scaled premise and DNA polymerase molecules attached to a single DNA strand and move along the strand extended the primer, forming new single strands of DNA called leading strand and lagging strand. The DNA polymerase that forms the lagging strands must synthesis discontinuous polynucleotide segments called Okazaki fragments. The DNA ligase enzyme then connects the pieces of the lagging strand. So many explanations from me. I hope it's useful for all of you. See you!